Nobody on that half of the room? What's an annotation? So an annotation is a description of the genome. It annotates the genome and describes where all of the genes are located in chromosome coordinates. So each annotation is specific to a genome. So you'll have an annotation of where genes are located in the human genome, where genes are located in the mouse genome, chimp genome, whatever your organism of interest may be. And keep in mind, as more information comes out, genome builds get updated. So we've been talking about working with HG19, which is a specific build of the genome. So when we were using STAR, we downloaded the annotation for the genome build HG19. And that is a GTF file that we got from GenCode. So again here, this program needs information about where to look for these genes. If you guys think about what it is that we're trying to do with feature counts, we're taking the reads. Right now, all we have is a BAM file with chromosome coordinates describing where all of these reads are located. But if we want to quantify gene expression, we need to give it an input, an annotation file that's telling it, okay, here's where all of the genes are located, and I want you to quantify the expression of all of the genes that are in this annotation file. Does that make sense? Okay, so when it's asking you for an annotation, is this something that you guys already have? Yes or no? Yes. Cool. So you should be able to use that already. Okay. And it gives you a quick description of single and paired end reads. So can this program handle paired end data? Yes. Great. Feature counts can be instructed to count fragments rather than reads. What's the difference between a fragment and a read? You can actually
The fragment is in data. You have two reads on each side. So part of that is understood where the computer is on each side and something that's in the middle of
Can you say that again louder? I didn't quite hear you. So the difference between a fragment and a read.
A read is a portion of it that is looked at whenever by the computer, and that is kind of of interest because it's a piece of the middle and it's
Okay. So I think you're kind of close. So when we made the library, remember we had this insert in the middle that we surrounded with adapters or whatever. Okay. So this is our fragment. It's called a fragment because when we made our library, we just started with these long pieces of RNA, poly-A tail. We reverse transcribed them into long pieces of cDNA, and then we randomly chopped them up because we can only sequence short bits. So these chopped up pieces are referred to as fragments that got put in between adapters when we sequenced. But when we did our sequencing, we sequenced paired end, meaning we had a primer that bound here and extended by 100 bases and then stopped. And we also had a primer that bound here and extended by 100 bases and then stopped. So this is what we were referring to as R1 and R2. Read 1, read 2. So these are the individual sequences that came off the machine, and they're referred to as reads. So for each fragment on paired end sequencing, we have 2 reads. We have read 1 and read 2. So when you're counting something, a lot of programs by default will just group these pairs together and call it one read and one fragment. They'll use it interchangeably. But the specification of what feature counts is telling you right here is that you can actually count based on fragments, and you're going to specify that to show that we only care about the quantification of the entire insert. If we were to be counting instead based on reads from paired end data, we would be double counting, right? Because this fragment has 2 reads associated with it. If we count on a read base instead of a fragment base, we're going to be double counting reads that are mapping to the same fragment. So that fragment would get 2 hits. and that's not what we want to do. Questions? Cool. So just reiterating that there, if paired reads are used, then each pair defines a fragment. So we're counting the fragments. Features and meta features. You'll notice as you start working through the flags, you can choose what feature you want to count to, and when you say feature, it means something like an exon, an intron, a gene, a transcript. So because we're quantifying gene expression, we're going to count based on genes. But if you were interested in how many reads we're mapping to each specific exon within a gene as opposed to the whole gene in general, you could use a different feature, and that feature would be exon. And you can see the options for that in the annotation below. This is kind of telling you how it is working, how it deals with, say, multiple overlaps, or how it deals with exons and introns. If you're wondering about the question of, okay, so if I have a spliced read or an insertion or deletion in the read, is it counting it twice? How is it handling all that information? Again, that's written out in pretty good detail here that you can look at further. Importantly, this is something that you guys should always look at. What sort of output is it going to give you? So it's telling you here, the output of this program is saved in a tab delimited file. So what's a tab delimited file? Yeah?
There are tabs between each column.
Tabs between each column. Good. So when we look at something, each column is spaced away by tabs. So each of the columns are going to be gene ID, the chromosome position, start, end, strand, and length. And then it's going to put all of these things into each of those columns. It's going to tell you here that it also includes status info of a summary. So you're going to get an output file with the counts of the reads that went to each gene. You're also going to get a summary, and in the summary it's going to report all of these different features, and then they give you an explanation for what those features do. Okay. And ultimately we're going to get to this program usage and all of the different options that you can use are reported down here. Okay, so this time, instead of actually writing the whole command, what I want you guys to do, again, working with the people around you, just go through here and tell me which of these flags you're going to use. So go through each one of them and decide as a group, yes or no, are you going to include this in your command? I'm also going to point out for you guys, because I don't like this documentation quite as much as other programs that we've used. It can be a little bit hard to read. So if you go back to your, when you Google the feature counts user manual, that first link is actually pretty helpful because it can show you some example commands down below. So you can get an idea. They don't do a very good job in the documentation of telling you which things are required and which things are optional. But if you want some examples, you can take a look here to see how other people have been running the program. And then go back through and decide which ones of these are important for us.